Hi, my name is Janelle, and this is me explaining adaptive immune response. So it starts with a pathogen, and in this pathogen, the innate presenting cells usually detect the pathogen and presents the antigen to the adaptive immune cells. Then the macrophages and dendritic cells will take up any pathogen through phagocytosis. As a part of this phagocytosis, the pathogen will be degraded, and after the degradation, pathogens are presented on the surface of the cell. This allows them to be detected by the adaptive immune cells. Um, they are presented um, on special molecules called MHC, and there's two different types. There's MHC1 and MHC2. There are CD4 plus T cells and CD8 plus T cells. Both of them have T cell receptors, which are protein that are on the surface of the T cell that detects antigens. The CD4 plus pairs with MHC2 and CD8 plus pairs with MHC1. If both are able to bind to the antigen and the antigen presenting cells are sending out activation signals, both of them will become activated. When CD8 plus is activated, it turns into a cytotoxic cell, T cell. It patrols the body and looks for cells that are infected with a pathogen with the antigen that is between the MHC1 and the TCR. When the antigen is found and a part of a body cell, that means that the body cell is infected with the pathogen. All the cells in our body have MHC1 that are able to present all of the molecules that are in the cell on the surface. So this means that if there's a pathogen present in the cell, they will also be presented outside of the cell. The antigen matched to the T cell receptor on the cytotoxic T cell will trigger a response. The cytotoxic T cell will release chemicals that cause the infected body cell to die. So cytotoxic T cells target intracellular pathogens for destruction, opposed to the MHC2, which are going to target the extracellular. Now onto CD4 plus cells. When it's activated, it becomes a helper T cell. Oftentimes, the T cell aids the B cell and helps activate them. B cells make antibodies, some that are put on the surface of B cell receptors. If the B cells come into contact with the pathogens, then the antigen will bind to the B cell receptor and the pathogen will be internalized. The pathogen will get degraded and presented on the surface of the B cell. The MHC2 will display the antigen, and the antigen will bind to the TCR on the helper T cell. The helper T cell will activate the B cell, which then becomes a plasma cell. A plasma cell will pump out massive amounts of antibodies into the extracellular fluid of the body. The plasma cell is often called an antibody factory. Then the antibodies encounter the pathogen with the antigen, they will bind to it. This targets the extracellular pathogen for destruction.